RNAI group, and she has been associated with the Biosafety Committee for the past 7 years and currently heading the Biosafety Committee. So may I request ma'am to kindly guide all of us about this very important topic on biological safety.
Yeah. So, good afternoon and welcome to this next session on soft skills. And today's lecture will be on the foundation training in biological safety. So, I can start with the first question as to what are biohazards and why are they important? So, when we say biohazards, these include all the hazards that are involved in a bioresearch. These would include the procedures and the practices followed in the lab, and would also be the samples, mainly the pathogenic samples or the mammalian tissues that are being used as research materials. And who is the major risk group? This would be the researchers or all the lab personnel who will be handling or manipulating these cells, and because they are at a risk of exposure to these potentially infectious pathogens which may be present in the tissues or the cells. There may be risk of exposure due to any procedural or handling errors that may happen, and mainly because of the pathogens which are present in these various samples, or due to the risk factors associated with the various procedures and the practices. So, this brings us to this very important aspect of understanding biosafety, and this is important because what we need to understand that what we are trying to save you or protect you from. It is from all kinds of injuries, accidents, and anything which leads to accidental spread that has to be quickly contained and restricted to that area within the lab. And this can be done by being a little aware, following all the safety principles, and as far as tissue handling is concerned, sticking to the standard microbiological techniques for using within the facilities so that the risk level can be contained and there is no risk from the pathogens that you are handling. So most of it, the basics of these injuries and accidents are covered under good lab practices, and we believe that most of you are already aware or trained in this. So we focus for today's talk. We focus only on the microbiological and the other biological techniques and the safety issues associated with them. So for all the Indian labs, we have a set of guidelines which have been published by the RCGM through the Department of Biotechnology, and this is our guidebook that we need to follow as far as the principles of biological safety are concerned. But what we need to understand is the requirements on which the biological safety or the parameters on the basis of which the biological safety parameters are assessed. And the primary among themselves is assessing the health risk. Once the health risk is assessed, then appropriate control measures or preventive measures need to be adopted to prevent or to control the exposure. Use all kind of control measures in the facility to minimize the risk of exposure and spread. Each of these should have a lot of standards or test control measures which can be maintained and examined regularly to ensure that safety is in place. The most important parameter again becomes individual health surveillance, both before and after the experimental procedures are being conducted. So this needs to be done at regular intervals. Information, instruction, and training of the personnel forms a very important part of this biological safety, because once you are aware, then you can adopt the appropriate control measures or preventive measures to minimize exposure. And of course, labs should or facilities should be equipped to handle or deal with any accidents, incidents, or emergencies that may happen to minimize the risk events. So before going into that, we need to understand what are the hazards. The hazards here would be all kinds of biological agents, like I mentioned, either the pathogens or the tissues that we are handling, and based on the degree of pathogenicity, that is the increasing hazard to human health, they have been classified into 4 groups. Group 1 contains all those which are unlikely to cause human disease and are considered to be relatively safe. Group 2 would include all the agents that can cause a human disease, but it is unlikely to spread to the community. There could be an individual infection, but this is usually, and there is usually a treatment available, and there is a very minimal chance of it to spread to your neighbor. Group 3 would be a little more higher in the hazard group and would include all those cases where a severe human disease can develop and there is a chance that it can spread in the community, but an effective line of treatment is available to control or cure it. And group 4 will include all the human diseases that can cause severe infections in the humans. They have a higher chance of spread within the community, and possibly an effective prophylaxis or treatment is not available to totally cure the disease. So based on these parameters, the biological agents have been classified as group 1, group 2, 3 and 4. Now, when we talk about the hazard group, then we have to talk about the containment level or the laboratory level which is required for doing experimentation on the pathogens in these hazard groups. Experiments are very important because unless you research and come up with the line of treatment, how will we find a cure to it? But important here is to handle whatever you are doing safely so as to prevent risk to ourselves as well as to prevent the spread. And to deal with these, there are specific containment levels which have been identified and which have been described. So, based on the hazard group, there is a corresponding containment level. So, thus we have 4 containment levels: 1, 2, 3, and 4 for hazard groups 1, 2, 3, and 4 respectively. Now, to handle the experimentation or the containment level for each hazard group, there is a specific design of the facility or the research facility that is required. So for risk group 1 and for major cases of risk group 2, a basic laboratory structure or a facility design is sufficient with certain requirements to prevent spread and to prevent infection. For risk group 3, there is a higher level of containment. So these are called specific containment laboratories or the special facilities which go by the term as BSL 3 facilities. And for the hazard group 4, then you have the maximum containment facility or the BSL 4 facility to do the work with these experiments. Now, how do we recognize what containment level is required for a particular requirement? And this has been very well defined in the form of 6 P's. These 6 P's can be categorized into 2 groups of 3. The first one to assess the risk, and the second category to manage the risk. Now, the risk assessment would include the 3 P's associated with the pathogen, procedure, and the person. Pathogens, that is the biohazard which is being handled, the procedures or the manipulations or the experimentation that is being done to study or to research on this particular biohazard, and then the personnel, that is the people, the scientists, the technicians, and the other lab people who are involved in handling the biohazard. And based on these three P's, the risk is assessed and the appropriate management practices are come into play. And what are these? This would involve the safe practices, that is having a safe laboratory area and a proper procedure defined to handle the pathogen, have protection, that is this maybe in terms of protective clothing or safety equipment that have been used, and then overall place, that is the work location or the research facility where the experimentation or the manipulations are being performed. So once we define or we understand the three P's involved in risk assessment and the others required for risk management, an effective biosafety environment can be built. Now going a little further to it, now we have defined the risk group and the containment levels required, and synonymously the terms biosafety levels are used synonymous to the containment levels. So again there will be the 4 levels, 1, 2, 3 and 4 for risk group pathogens 1, 2, 3 and 4. And what are the primary determinants which will define which level of containment has to be put into practice? This can be understood if we understand the infectivity, both the disease and the nature of the pathogen, the severity of the disease which they cause, whether it can be treated, if it is curable or non-curable, the transmissibility, that is the time it takes, whether it spreads or not, and if it spreads, then the time it takes for the transmissibility, the nature of work being conducted, whether only a small part of the pathogen or the pathogen per se is being worked upon. Then the origin of the microbe or the nature equation, and then the most important being the route of exposure, whether it is through contact or through the air or other means. So based on these parameters, the biosafety levels can be defined and appropriate levels are put in place to do the work. And wherever there is some confusion on the biosafety levels, it cannot be accurately determined. It is always better to work at a higher level of containment so as to protect oneself as well as the neighbors in the lab. So, going forward on the evaluation criteria for these pathogens, the first and foremost is to identify the biohazard, the way in which the agent can harm the human health. And that would, once this is identified, the appropriate containment level required for its handling can automatically be adjusted. The second thing after identifying the hazard is the nature of the work that has to be done. Whether only a particular gene or protein or a part of the pathogen needs to be understood or characterized, or the experiments would involve handling the entire pathogen or the screening for the pathogen. All these features would include the nature of the work being involved, and based on these we evaluate the risk factor. That is, what is the likelihood or the severity of its causing the harm. Once the risk factor is evaluated, then appropriate safety measures can be employed, whether it could be the level 1, 2, 3, and only in specific areas the BSL 4 work is done. It is not carried out in all kinds of research laboratories. So once the safety levels are put in place, the work can begin. But the most important point or a parameter here, which is normally overlooked, is to record and review the assessment. It is a constant monitoring and a constant assessment of the risk factor of the lab premises or the facility premises, as well as individual health, becomes a very important factor in this evaluation criteria, and at times can help in evaluating whether the risk group needs to be upgraded in specific conditions if required. So, we can summarize by saying that the safety measures are defined by the work requirements and the pathogens being handled, and based on once the containment level has been identified, then appropriate safety measures can be employed. But what do we need by safety measures? It's a very broad term, and this would include all kinds of procedures, that is the handling as well as the biological procedures, the use of safety equipment and use of protective clothing, wherever required, especially in case where pathogens of group 3 or group 4 are being handled, then a specific design of the facility is required so that the pathogens do not spread or leak out of the facility either through air, water or any other route, and have an appropriate monitoring system in place to ensure that safety measures are in place and any kind of spread or infection is not happening. So their monitoring plays a very important factor. So safety measures, when we talk about safety measures, it encompasses all the 4 features that combine to make the laboratory safe for doing a specific kind of research. So, like I said, the biosafety levels are of 4 kinds. The biosafety level 1 is the least hazardous group, and 3 and 4 are the high risk group. So, generally, a lot of attention or focus is given to the work and as well as the personnel who are working in dedicated facilities for doing BSL 3 category or BSL 4 category of work. And normally, there is very little risk factor required with the BSL 1 containment or the experiment. So the most neglected happens to be the biosafety level 2, because we tend to take it in parties. Because the moment you enter one enters the BSL 3 facility, all kinds of instructions, SOPs are in place. So one tends to be more careful. But the slack normally happens in the biosafety level 2, and this is the so I decided to focus biosafety level 2 for today's talk, because this has to be a multi-module course. So as a first module, I thought to cover information on biosafety level 2. Now this is a moderate, pathogens having moderate individual risk and very low community risk. And if we talk about microbes, some examples can be the hepatitis B, HIV, salmonella, so on and so forth. These microbes, these are the microbes which can cause diseases in humans, in animals, even in plants, but with various severity. Not the examples mentioned here, but now I'm talking about the group 2 per se. And these would include the microbes that can cause a disease in any kind of organism. They are typically indigenous and normally have a moderate or a low level of spread within the community. And the laboratory exposures, if any accidental exposure may happen, they can cause serious infection, but effective treatment or preventive measures are available to handle these exposures, and as I said, the risk of spread of infection is limited. Now, since these microbes do not spread but can cause serious infection, so prevention is better than cure. So we need to have specific guidelines or safety precautions in place so that the risk of this infection can be minimized, if not removed altogether. Now, so what kind of tissues or what kind of work needs to be carried out under BSL food containment? And broadly, if we say, it will be appropriate that work is being done with any kind of human-derived blood or blood tissues, or body tissues, fluids, cell lines, or anything which has the potential for the presence of these risk group 2 microbes or infectious agents which have potential pathogens for humans. All kinds of oncogenic viral systems also get classified as BSL-2. The recombinant DNA activities, including studies which involve the construction of transgenic animals or transgenic plants, get categorized under BSL-2. All non-recombinant cell or tissue culture systems on which are being used with the risk-2 microbes are handled under or categorized under BSL-2, and all high volume or large-scale activities with risk-2 organisms also get categorized under BSL-2, so that the appropriate safety measures can be put in place to prevent their spread or contamination in the local environment. So, having said that, I bring you to this document which is available or which has been issued by ICGEB, and where they list the microorganisms, the animals, plants, insects, and even aquatic animals which get categorized under the risk group 2 and require biosafety level 2 kind of containment facilities. And other than these, there is a whole list which is available in the document. But other than this, all the genetic engineering experiments which involve use of or pre-approved host vector systems in which a gene is inserted, and if this gene is a pathogenic determinant or is capable of causing any infection or any harm, then it gets classified as BSL-2. If there is any non-approved host or vector system for which data is not available, whether it can cause any damage or any kind of injury to the organism, and then it is appropriate to handle them in BSL-2 facilities or lab. Use of replication defective viruses as hosts or vectors is included in this group. All, as already mentioned, all risk-2 microbes, whether they are infectious, defective, or even heat-killed, when being used in the presence of viruses, come under BSL-2 containment. DNA from recombinant, when the whole DNA, the whole genomic DNA from any of the risk group 2 or 3 organisms is being used either to transfer to the non-pathogenic prokaryotes or to lower eukaryotes or even single genes which may be pathogenic, get classified under group 3. So, other than that, maintenance or experiments with animals or plants which are infected with group 2 organisms also fall under this category. Experiments with GE animals, that is these genetically engineered animals or plants, which contain pathogenic determinants or whole genome of the RG2 group of microorganisms, need to be handled under BSL-2. In case of plants, all genetically engineered plants which confer herbicide tolerance or pathogen resistance need to be handled under group 2 containment facilities, because we don't want these genes getting transferred in the environment outside. And in specific cases of gene editing which involve SNP2 and 3 types of modifications, get also classified under this group 3. So, now we know that the pathogen or the infection causing host and microbes get classified as BSL-3 and 4. I have also listed in details all the criteria of all the experiments which get categorized under category 2. So what is not included, all which doesn't get included in the group 2 list and is not pathogenic, gets classified as category 1. And it is very important to understand what is not included, because there is a thin line when we are working in the labs. There is a very thin line which categorizes these experiments as 1 or 2. So, just to understand the risk group 1, wherever experiments are being done with approved host vector systems in which the donor DNA is originating from risk group 1, organisms. These are not to be included in RG2, but they get categorized as risk group 1. All these insertions or single genes which do not have any adverse effect on the health or do not give rise to any infectious agents are risk group 1. The self-cloning vectors, there will be infusions of protoplasts between non-pathogenic organisms, breeding animals, even if they are genetically engineered animals, maintaining or housing them, then specific animals which do not contain any pathogenic determinant can be handled under the biosafety level 1 containments. And the SDN1 type of genome editing experiments also get grouped here, but these need to be, the categorization needs to be reviewed by the institutional biosafety committee to guide or to help in classification of a particular experiment as category 1 or category 2. So for category 1 experiments, only an information to IBSC is required, but all category 2 experiments require prior approval from the biosafety before starting the experiment. So having said that, we need to understand what are the safe lab practices that get employed under the BSL2 facilities. So apart from the good lab practices which I mentioned earlier and which are the basic lab requirements or etiquettes of working in the lab, other than that there are specific guidelines which have been defined for a standard or a regular BSL2 research lab, and these include following all the routine microbiological practices of starting the cultures, using the cultures and then decontaminating the cultures and appropriate waste disposal. All these standard microbiological practices get on the baseline. Work can be performed on an open lab bench. Wherever there is a chance of getting an aerosol or a splash or any kind of toxic fumes or toxic elements, then it is better handled in a hood or a biological safety cabinet. Access to BSL2 laboratories needs to be limited or restricted, even if work is not being conducted or the lab is not fully functional. The access should be limited. It is not an open access area where you can freely walk in and walk out. Walking into a BSL2 lab, or rather before walking into a BSL2 lab, the personnel should be equipped with the safety clothing, which would include the PPE kits, a normal lab coat, having hand gloves, an eye protection or a face shield as and when required in specific cases. It is better to perform work in the biological safety cabinet. There should be a designated area or a bench or a corner where personnel can come and store the boxes, the samples which are being received or handled, and this specifically refers to the human tissues like blood and the other body tissues which are coming for further analysis. It is preferable to have a designated area and to follow the basic practices of decontaminating the area before and after use, and to employ all the safety precautions for proper handling and waste disposal. In specific labs where pathogens, work with pathogens, that is the risk group 2 pathogens is being carried out, it is appropriate to have biohazard warning signals at the access points on the gate, to do appropriate immunizations or tests for the agents handled in the laboratory, and have well-defined SOPs on the door so that the personnel are aware on the kind of routines or the procedures that have to be followed within the lab. So a smaller area or a smaller corner in the lab can be further restricted or defined so that specific handling takes place within that that area and the chance of spread is further minimized. So the primary, other than these, the primary concerns which are included in a BSL2 facility include accidental needle sticks or cuts or any kinds of mucous membrane exposures which may result in an accidental infection or contamination. So it is, and this is where SOPs become important, because the personnel or the lab workers should be familiarized with all the written instructions for the laboratory procedure, and they should know the proper responses to any kind of emergencies. If there is a nick or a accidental nick or a cut, then what kind of safety procedures need to be adopted. It is better to have a first line of safety equipment, including lab coats and gloves. Whenever transporting materials across laboratories, it is better to use leak-proof secondary containers, so that in case of an accidental fall or a spill, it is contained within this container and is easy to be handled. It's better to wash hands after handling the biological materials. Washing hands before and starting work and after completing the work is an essential basic etiquette, and it is better to wash hands, gloves, put on gloves, do the work, and then remove the gloves, wash the hands before leaving the area in that order. Eating, drinking, smoking and other activities should not be done within the research work area, because it can be a root cause of accidental exposure, which also you are bringing in accidentally maybe bringing in some pathogens or unwanted materials in the lab which are not required. As far as handling is concerned, again now the good lab practices come into place. So mechanical pipettes should be used as far as possible. Do not use sharps. There should be appropriate mechanisms of disposing sharps in case there is any plastic breaks or plastic breaks or beaker breaks. Then the set procedures for collecting the glass pieces and decontaminating the area need to be followed. There should be procedures, laboratory procedures in which there is a chance of creating a splash or a spray, especially when you are opening a small Pendoff tube or a microfiche tube. All these and when there is a pathogen involved, it is better handled within a biosafety cabinet. Now, the biosafety cabinet is placed in the lab, and as well as the other safety equipments need to be constantly monitored and certified and maintained, so that the safety levels are maintained. So, monitoring again of the lab equipment itself becomes a very important factor here. Extreme precautions should be taken when handling sharps. I've already talked about it. You need to decontaminate the work surface daily before and after beginning the work. Decontamination of waste materials before disposal is a very important procedural step and must be followed rigorously. There should be an effective insect or rodent control program so that you don't have any unwanted guests in the lab. And of course, spills, exposures, illnesses not only need to be taken care of, but also need to be reported, so that appropriate follow-up and recording can be done, and these records can be maintained. Other than that, a BSL2 facility needs to have at least a sink and an eye wash area, so that a splash and water should be available for an easy splash wherever required. The doors, the entry, as I said, needs to be restricted, so it is better to have self-closing doors, and within the laboratory also dedicated areas or spaces, if the space allows, can be created to separate the working space from rest of the facility. The laboratory bench area should be easily accessible for cleaning. It should not be piled on with books and journals and papers and other stuff, and whatever is being used, the surface should be easily accessible for cleaning. And having said that, the bench tops need to be impervious to water and it should be resistant to acids and alkalis, otherwise the whole purpose gets lost. Windows, if any, even in the lab, need to be fitted with privacy and are best closed at the time when experiments are in place. And there should be an effective this area or designated area or an autoclave should be available, as well as alternate methods of decontamination where you use specific chemicals and other detergents are available for ensuring that effective decontamination is happening and proper disposals are in place. And over and above these requirements, like I mentioned earlier, surveillance becomes a very important factor, and this is surveillance for the working of the safety equipment, surveillance for health of the employees in the lab, as well as recording of any illnesses and absences so that it can be called upon. And based on the kind of work, there are specific BSL2 facilities that are also available in institutes like ours. So there may be these, what we talked about was the general research labs, but then there are specific facilities like the animal BSL2 facility for handling the animal experiments, and here again the animals that are inoculated with risk group 2 microbes, category 2 GE experiments, or animals which are genetically engineered animals which are harboring the risk group 2 microbes for whatever experimentations can be safely handled. Similarly, there can be specific facilities for plants and arthropods which can be designated as the plant BSL2 facility or the arthropod BSL2 facilities, where specific work appropriate for that particular organism can be carried out. And here just a brief mention that about some of the gene editing experiments, specifically those which are involved in group 2 category, need to be carried out, and it is best to start the experiments of the which can get classified under group 1 also within the BSL2 facility, and once the organism, the gene edited organism or the construct is ready and is screened, screened and tested to ensure that it falls under group 1 category, then it can be handled under the biosafety level 1 conditions. So not everything is all the instructions are for the personnel. Also, there are some additional responsibilities of the leaders, of the group leaders, of the PIs in the lab, because it is their duty to make initial determination of the proper physical and biological containment levels that are required for the project, ensure that necessary experimental protocols are available, and these are in accordance with the guidelines that have been issued by the biosafety committees or the recombinant gene group committees. Wherever required, seek permissions of the IDSCs and RCGMs before starting the work. All BSL2 categories require prior permissions from IDSC, whereas 3 and above require permissions from RCGM for beginning the work. Ensure that no work gets initiated unless appropriate approvals are in place. And in case of Indian labs, we will say that they have met all the requirements as per the DBT guidelines. There should be strict instructions to the laboratory staff about the practices and techniques which are required to ensure the safety and procedures. The SOPs, specific SOPs, if any, need to be listed and need to be displayed, visibly displayed as an easy access point. And wherever required, there should be a provision for recording the health records of the laboratory personnel. And if there is in case of any incident or accident, so that appropriate follow-up can be done. So, having said that, details on what needs to be done in the facility, what should be the facility guidelines or the design strategy, everything for us is mentioned in this laboratory manual, the guidebook, which has been efficiently compiled by DBT and the RCGM, and this is the book to fall upon in case of any queries or any uncertainties that may arise. So I will conclude here, and the take-home message of today's talk can be just summarized as one line: is work safely, stay safe, and do your experiments properly, but never experiment with safety. Thank you.